So today we are going to discuss about the polymorphism. What is polymorphism? Let's discuss about it. So polymorphism is of two types. One is the genetic polymorphism and one is the DNA polymorphism. So, uh, so genetic polymorphism is a broader term. It includes the let me just write it down. It includes three kinds of polymorphism. Number one, you can say DNA polymorphism. Number two, there is epigenetic polymorphism. Number three is the regulatory polymorphism. So, genetic polymorphism is a broader term, while the DNA polymorphism is a smaller term, you can say, the briefer term, that will include only the DNA sequences. So, if we see here the epigenetic changes, if we talk about the epigenetic changes, like for example, if you have two individuals, individual 1 and individual 2, these two individuals have, let's say one gene P53. Both of these individuals have the same sequence, but their methylation pattern is different. Maybe instead in this particular number one patient, the sequence is CG, but in case of the number two individual, it could be CG, but this C is methylated. And when you know the particular sequence is methylated, it basically suppresses the expression of gene. Okay, so here you can see there is no difference in the sequence. So the point to be noted is no difference in the sequence, only the methylation pattern is different. No difference in the sequence. Okay. Here you, if you see here now the another thing is regulatory. Regulatory regions include promoter, uh, silencers, enhancers, the sites that bind to the transcription factor. So what happened if there is any polymorphism in these regions? Because this is again not the DNA polymorphism. So if suppose if particular gene has a strong promoter, then the expression of that gene is going to be high. But suppose in the another individual, if this promoter is weak, maybe you will not get the expression of that gene high. Maybe there is a low expression, but the gene sequence is same. Only the regulatory regions are different. And because of this, genetic polymorphism is considered as a broader term. It includes DNA polymorphism, it includes epigenetic polymorphism, it includes regulatory polymorphism, and also because of these polymorphisms, there is a variation in the phenotypic trait. How? Because if suppose if this particular gene expression is high, maybe the eye color is going to be uh, black. If the expression is less, maybe the eye color is going to be brown, like that. Phenotypic traits could also be different because of this. So this is regarding this particular sentence. So genetic polymorphism basically includes a DNA polymorphism, but also refers to the variation that is not only sequence based. One is epigenetic, another one is regulatory, and because of these changes, there is a variation of the phenotypic traits. So genetic polymorphism include DNA polymorphism, epigenetic polymorphism, and the regulatory polymorphism. So genetic polymorphism, it confer diversity definitely in the population, and that is why it is used to study the population diversity, migration patterns, and relationship between and within the various populations. And basically it is of two types. One is the balanced type, another one is the transient type. Coming to the balanced type, if we talk about, in the balanced type, both the alleles are present. the homozygous, the dominant one, and the recessive one. So this is the condition of the balanced polymorphism. Why it is called balanced? Because both the alleles are present. If this is the case, if the polymorphism is present in both the alleles, this is very important because this provides a certain sort of advantage. For example, if you see the person like that has the sickle cell trait, okay, so because of this sickle cell trait, these patients, these individuals basically, because these are not patients, these individuals are resistant to the malaria parasite. So these individuals are resistant to malaria parasite. So this is an advantage. If both the alleles are present, it means they are having a sort of advantage to reproduce them or to survive better. So this is the balanced uh, sort of genetic polymorphism. Now you can see the definition here, whatever I told you right now. Balanced polymorphism is characterized by the presence of both of alleles, means capital A, small a, dominantly small a. Both the alleles are present rather than having a two copies of a single allele alone. This type of polymorphism is present in the heterozygous conditions and brings the heterozygous advantage. Individuals that carry the both alleles may be considered more fit, are able to survive and reproduce as compared to those they are having two copies of the either form. And the example is the sickle cell trait, as I mentioned here. So, Elliotson in 1954 reported that the individuals which are heterozygous, they have the selective advantage of being a malaria resistant, means they are resistant to the malaria parasite. Therefore, the heterozygote advantage of the sickle cell red blood cells provides a protective effect as the malarial parasite cannot grow in these cells. So that is some sort of advantage. Now coming to the transient polymorphism. And again, remember, we are talking about the genetic polymorphism. Now coming to the transient polymorphism, both the alleles, suppose they are present, then one allele is replacing the another allele. So one allele is completely replacing the another allele. Okay. And very good example of this is the moth. Okay. So you can see here when the progressive replacement of one allele by another allele of the gene, the rare allele starts gaining an overall advantage, thereby spreading in the population. This will reduce the frequency of the normal allele. Here, one of the alternate forms of the allele is progressively replaced by the another allele due to the inheritance. Now it will pass to the another generation like that. And why this happens? Uh, this happens under the influence of the strong environmental selective pressure, which eliminates one of the alleles from the gene pool. Means if there is environmental pressure, one allele is completely going to be replaced by the another allele. And generally, this allele is the rare or the mutant allele. And if you want to see the good example of this is this, and here also you can see, although the process takes several generations, it is considered transient because it's not a stable one. The population is not maintaining both alleles in the long run. One allele is going to be replaced by the another allele, and this is known as the fixation. The very good example, if you see, is the moth. This is the moth, and this is peppered moth, or scientific name is Western Vetilaria. So this moth was having the two alleles. Let's say allele A and A, but after, because this was living in the industrial area, so industrial area industrial area is generally, dark in color. So because of that, what happened, This mutant allele, which is the small a allele, completely replaces the wild type allele. Suppose this is the mutant allele. This mutant allele completely replaces the wild type allele. Okay. And why it is doing this? Because of this mutant allele, the color of this moth becomes black in color. Suppose this moth, which was having the capital A small a, was white in color, but because this A allele is replacing the capital A, now this moth become black. So if you see here, the white moth or the black moth, in the white moth you will say capital A small a, but in the black moth you will say only the mutant allele. So because of this mutation, these moths become black, as you can see here also. This is the black moth. And what is the advantage of this becoming black? Because the surrounding insects cannot find and identify these moths because the industrial place is also black. They are unable to differentiate between the trees of that forest or that industrial region and the moth, and that is how they protect themselves. In case of the rural area, this becomes in the opposite. So why this becomes black? So that it can protect itself from the other insects and it can live a longer life. So this is a very good example of the transient polymorphism where the mutant allele completely replaced the wild type allele. Means the small a allele completely replaced the capital A allele. Now we are talking about the DNA polymorphism, which is a shorter term than the genetic polymorphism because it includes only the DNA sequence. Now, what is the difference between the DNA polymorphism and the mutation? DNA polymorphism you can find in approximate 1 to 2% of the population. That is, if it is like uh, less than that, then this is not polymorphism. So this is the difference between the DNA polymorphism and the mutation. And DNA polymorphism could be single nucleotide polymorphism, which could be then variable number of tandem repeats, mini satellites, microsatellites, then transposable elements, copy number variations. So we are going to discuss how many types of DNA polymorphism is there. Number one is the single nucleotide polymorphism. As you can see by name only, there is a difference in the single nucleotide. Okay, others are same. And it could occur because of the substitutions, insertions, and deletions. SNPs are prominent sources of variations in genomes, human genomes, and serve as excellent genetic markers. These single nucleotide polymorphism, they could occur in the non-coding region, in the coding region, but mostly they are in the non-coding region. So the effect is not that very high, but sometimes polymorphism, if it is in the non-coding regions also, they may lead to the disease. And the good example of this is, if you talk about is the beta thalassemia. Beta thalassemia, you can find a mutation in the non-coding region also. Okay. Again, some regions are very sensitive to the SNPs and some are not. For example, if we talk about the mitochondrial DNA, that is more susceptible to the polymorphism because that is near to the reactive oxygen species that are produced in the mitochondria. So mitochondrial genome is susceptible to the SNPs in compared to the genomic DNA. Mostly they are located in the non-coding region, so there is no direct impact, but sometimes if that is in the non-coding region also, they can lead to the beta thalassemia like disease. And how commonly you find after every 300 nucleotides, you will find the polymorphism. Okay, which means you will find approximate 10 million SNPs in the human genome, and human genome is 3.2 billion base pairs long. So 10 million bases are containing the SNPs. Okay. This is the thing which I was telling, that mitochondrial DNA is more susceptible because it is due to the proximity of the reactive oxygen species that are generated in the mitochondria. They can be present in the coding regions and the non-coding regions. Always remember, SNPs are very much common in the non-coding regions, but if it is present in the coding region, they are of two types: synonymous, where there is no change in the amino acid sequence; non-synonymous, when there is a change in the amino acid sequences. You know, every amino acid is coded by multiple uh, one amino acid is coded by approximately 3 codons, right? Only the last base, suppose if the amino acid is coded by A, AUU, AUC, AUG. This is just an example. I don't remember the code of the amino acid. But if this is the case, you can see all of these amino acids have the two base pairs which are same AU, AU. Only the last base pair is changed, but they are coding for the single amino acid. If the polymorphism is in the U region or C region or G region, then the amino acid is not going to be changed, and this is the synonymous. So this is the single amino acid we are going to be formed. So what does it mean? All of these codons are coding for the same amino, coding for same amino acid. Okay. So if there is any polymorphism in the, let's say, any region, C region, and it becomes T, then no difference in the amino. In case of the non-synonymous, there is a change in the amino acid sequence. It could be missense, where the amino acid is going to change the protein product. In case of the nonsense mutation, you know, the nonsense codons are three types: ochre, amber, or boy, which is UAA, UAG, and UGA. These are the stop codons. If any polymorphism is creating these kind of stop codons, then the transcript is going to be shorter. And if it is shorter, then the protein is going to be shorter. Suppose the normal protein is 70 kD kilodalton, let's say normal protein. The truncated protein could be 35 kD, let's say truncated protein. So this protein cannot function like the normal protein. So these kind of mutations are non-synonymous SNPs are very important because they change the behavior. So the non-synonymous SNPs are two types, missense and nonsense. When the change of the single base pair causes the substitution of a different amino acid, it is known as the missense mutation. And if we talk about the nonsense mutation, amino acid substitution leads to the protein may cause to be the protein non-functional. In genetics, a nonsense mutation is a point mutation in a sequence of DNA that results in a premature stop codon or a nonsense codon, producing truncated, incomplete, and generally non-functional protein. As I told you right now, the normal protein could be 70 kD long, but the mutated protein or the polymorphic, due to the polymorphism, the protein that is going to be produced is, let's say, 35 kD, then that becomes non-functional. Now coming to the insertion and deletion polymorphism. That was a single nucleotide polymorphism. Now we are talking about the insertion and deletion polymorphism. They are basically what happened, known as indel. In short, they are known as indel. Okay. In means insertion, del means deletion. And how it is caused? Because How this is going to be caused? This could be because of the strand slippage. The mechanism that leads to the insertion and deletion is the strand slippage. Let's say this is your template strand and it is having the CA, 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 and CA. Now this is the template strand. This is going to be your 5' to 3' direction. Now this is the coding strand that will be having TG like this. This is again 5' to 3' direction. Now what happened? In case of the repeats, what happened? This particular GT that forms the loop, there is a strand slippage and that forms the loop. So now you will be having CA, 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 and now you will be having GT, 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 and one GT is in the loop. Let's say this is the loop and this has the GT. Now what the DNA polymerase will do, so this is step number 1. So because of the strand slippage, what is going to be happening? This is the strand slippage. So this strand slippage is forming the loop now. Now because this is the loop, now the DNA polymerase will add GT here. Okay. So now how many GTs you have? 6 GT. 6 GT. 1, 1, 2, 3, 4, 5, 6. Instead of 5 GT, you are now having the 6 GT, and this is known as the insertion polymorphism because there is an addition of the GT. Now this particular sequence will serve as a template strand. Okay, and then you will be getting the permanent CA insertion in your genome. Okay, now how come this is regarding the insertion, how the deletion will happen. So the important point that needs to be remembered in this case is strand slippage is occurring in the coding strand. So if I write the strand slippage as S, then you can see the SS is going to be in the SSN strand slippage in the coding strand. Okay, and that leads to the insertion. Okay, now coming to the deletion. In deletion, what happened? This is the insertion. In deletion, what happened? The strand slippage is in the template strand. So what will you get? CA, 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 and one CA will go in the loop. Okay. Now, DNA polymerase will make GT, 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 GT. It will not recognize this particular CA. This DNA polymerase will not recognize this particular CA, and instead of 5 GT, you are, how many GT you are having right now? If you see here, you are having 4 GT instead of 5. So that is deletion. So this is the deletion. This is the insertion. Insertion, there is a strand slippage in the coding strand. In the coding strand. In the deletion, strand slippage in the template strand. So insertion and deletion, the major region of the insertion and deletion polymorphism is always in the regions which are containing repetitive sequences. The another thing is uh, this happened because of the strand slippage. So now you can see insertion or deletion polymorphism are the variations in which a portion of DNA is added or deleted. It is just not one nucleotide, it could be multiple nucleotides also. Okay? If the insertion and deletion, which is not in multiple of 3, occurs in a protein coding region, because protein is coded by the 3 amino acids, it is also known as a frame shift mutation. Short region insertion and deletions can be formed by the strand slippage, and the deletions or insertions of the longer regions are formed usually due to the homologous recombination. Strand slippage I already discussed right now. So strand slippage occurs during DNA replication when a newly synthesized strand or the template strand temporarily detaches and misaligns upon allele. So if there is a strand slippage in the template strand, then it means DNA polymerase will not recognize that particular sequence in the loop region, and therefore there will be a deletion. If it is in the newly synthesized strand, there will be the insertion. So this misalignment results in insertions and deletions, especially in the regions with the repetitive sequences. And the frequency of insertion indels in human genome is 1/10 the frequency of the single nucleotide polymorphism. Uh, Usually they are smaller, means insertion and deletions are 90% are 1 to 10 nucleotides, 99% are 9% is 11 to 100 nucleotides, and 1% involves greater than 100 nucleotides. And this is because of the homologous recombination. 5 major types of indels you can find: just a single base pair insertion deletion, or a single base pair expansion insertion deletion, then multiple base pair expansions, transposon, you know that jumping DNA, and I have made a video on transposons. If you want, you can have a look on that. Transposon insertion, then the indels containing the random DNA sequences. Okay. Insertion and deletions usually are very common in the hotspot regions. One example is the angiotensin 1 converting enzyme. You know this enzyme is produced in the liver, and that converts angiotensin 2 to the 1. And because of this, there is a vasoconstriction, means there is a constriction in the contraction in the muscles, and because of this, there is a vessels basically. So the high blood pressure, that leads to the high blood pressure. So higher concentration of ACE is not good. And you can see there are two variants of the ACE. One is the shorter variant, which is producing the high ACE concentration. High ACE concentration means high blood pressure, because they will lead to the constriction of the blood vessels. And if it is longer, then that will protect it from the blood pressure. So polymorphism you can find in uh, like any gene. Okay. Uh, Show its effect on the phenotype. How the polymorphism is going to have an effect on the phenotype. If the polymorphism is producing the smaller allele, shorter allele, there will be high ACE concentration and high blood pressure, like that. Now coming to the repetitive DNA sequences, if we talk about if there is a polymorphism in the repetitive sequences, let's say polymorphic repetitive elements are DNA sequences that are repeated multiple times in the genome, with a number of repeats varying between individuals in a population. So one individual can have 50, one individual can have 500. A very good example is the muscular dystrophy. If you have 50 repeats, you are normal. If you have 500 repeats, you are not normal. This variation contributes to genetic diversity and can influence gene regulation, genomic stability, and even disease susceptibility. So, how many kinds of repetitive elements are there? One is the shorter tandem repeat STR, also microsatellites, 1 to 6 base pairs long. So, It can, one person can have 10, one person can have, number of repeat units, 15, like that. They are used in the forensic DNA profiling. One individual will vary from the another individual on the basis of these repeats. And also for the paternity testing that the child belongs to the same parents or not. So, depending on the search algorithm, there are approximately zero point 0.7 to 1 million locals, which are 2 to 6 base pair long in the human reference genome. Di and tetranucleotides constitutes about 75% of the short tandem repeats, while the remaining tri, tetra, they are only 25%. They are non-randomly distributed across protein coding regions, untranslated regions, and the introns. Simple tandem repeats, short tandem repeats containing dinucleotide repeats units that are much more abundant in the regulatory or the UTR regions than in the another genomic regions. In the coding regions of the genes, repeats mostly have either trimeric or hexameric repeat units, likely as a result of selection against frameshift mutation. The mutation rates of HTR often lie between 10 to the power 3 and 10 to the power 6 per cell generation, which is 10 to 10 to the power 5-fold higher than the average mutation rate observed in non-repetitive regions of the genome. So what this sentence means is, because these are the short tandem repeats, these repeats have high chances of mutation rate in comparison to the other sequences which are not present in the form of a repeat. It's quite high. The mutation rate is very high. Mini satellites, also known as the variable number tandem repeats, like 10 to 100 base pair repeats, and you can check these repeats if you are running them on the gel electrophoresis. Example is the telomeric repeats at the chromosomal end. Transposable elements, they can also have the polymorphism. They are usually 300 base pair long sequences, and I have made a separate lecture on that. You can go ahead and have a look on that. For example, is the allele polymorphism is linked to certain disease, specifically cancer. Then the copy number variations polymorphism. So what is the difference between the copy number variation polymorphism and short tandem repeats polymorphism? What is going to be there? These are the repeat sequences, generally 10 to 100 base pair, and you can say 2 to 100 base pair repeats. While in case of the copy number, either the whole gene is present in the, suppose this is gene 1, this is present in the second copy. This is the second copy of gene. Second copy of gene 1. Or either this is the gene 1, let's say, and this is the segment. This is present in the multiple copies. So you can say in one individual there is only one copy, in another individual this could have this particular segment in gene 1. If suppose this is gene 1 and this is the segment, this is the gene 1, and um, this is the gene 1, and this is the segment we are talking about. This segment here, this is present in one copy, and here this is present in multiple copies. Segment 1, if you talk about, this is present in 2 copies. So this is basically they are denoted as CNV, copy number variations. Okay? So that is the difference. It's not the repetitive sequences, either the whole gene. Here you can see You can see the whole gene is copied. This is gene 1. This is the second copy of gene 1. Here, only the segment of this gene 1 is copied twice. So in one individual, it could be in one copy, and another individual, it could be two copies. While in case of the repetitive sequences, that is always in the sequences are always in the form of repeats, usually to 200 base pair. Okay, so these are the copy number variations. The number of copies of a particular gene or genetic region is going to be differed. They are also known as the CNVs. It was normally consideration that all genes have always two copies in a genome. Recent discoveries have revealed that larger segments of DNA ranging from 1,000 to million DNA bases may vary in copy number from individual to individuals. Recent studies reveal that CNVs are 3 times the SNVs. It was found that the majority of CNVs were shared between human and chimpanzee, but in the human genome, approximately 1/3 of the CNVs are unique. To confirm these results were confirmed by another scientist, and other studies have also found that CNVs are also associated to the genetic diseases obvious in humans. So, very good example is cancer. In cancer patients, you can find the tumor suppressor gene may be present in high copies in the normal individuals, but not in the cancer patients. It is a very good example. With this note, this is the end of the lecture. Thank you.